Good afternoon, students. It is Wednesday, April 8th, about 3:05, and I'm glad to be with you again today. I've called most of you at least 3 times, and I've got a few more phone calls that I'm going to make this evening. But I'm trying to keep you current with all of the work, and that's the reason I'm calling you and emailing you and doing the neuro. So I hope you are staying current. Class notes, diagrams, your study guide at the end of neuro, and your videos will keep you current with what you have to know. Today we're going to look at the membranes of the brain. And now you'll know when you ever hear the word meninges and meningitis, you'll know what that means. We're going to look at these three membranes, and we're going to look at the blood-brain barrier, which is extremely important. It starts to build in our brain, that blood-brain barrier, right at the end of the first year of life. And because of it, it has saved our brain from toxins that would otherwise be destructive to the delicate neurons within the brain and the neuroglia. Okay. The questions that are pertinent in your study guide, I'll refer to as question 15, which is a discussion of the meninges. And I believe later on it asks you also what the blood-brain barrier is. Sometimes they just abbreviate that BBB. I'm going to start asking you some questions over the content of the material when I speak to you. So please study hard and don't disappoint me. I want you to answer those questions. Your reading in your textbook will be on 390 and 391. And you've got an excellent diagram of the meninges on page 390 and on 391. Thorough discussion in your textbook of the meninges. Okay. I'm going to give you a little list of videos to watch. I'll put this information on my email to you today, so you don't have to write it down real quick. I'll give you that information. I'd like you to read, and these are real short videos. Meninges on part 1, 2, and 3. These are on YouTube. And meninges and the skull. Those refer to the membrane, the three membranes. And I'd also like you to look at what is the blood-brain barrier, and in 2-minute neuroscience, the blood-brain barrier. So I'll give you that information again in my email to you, so you know exactly what we're supposed to be doing. All right. In the brain, you have 3 membranes that surround and protect your brain. And these 3 membranes from the outside going in, they are the dura mater, which means hard mother in Latin. Dura means hard, mater hugs them very tightly, is the pia mater, which means soft mother. So from the outside going in, dura mater, arachnoid, and pia mater. Now, I'm going to show you kind of a little rough drawing before we go over this illustration again, and another one that's in your textbook. And I did this, it's kind of a crude drawing because I wanted to see if you could if I could just illustrate it on a real simple layer, or show you these layers very simply. Now, this would represent the outside of the brain. These structures right here would represent the gyri and sulci, gyri and sulci. And I've got 3 colors here to kind of illustrate, I'm holding it just right so I can see that you can see it, the 3 layers. The dura mater I've done in a heavy dark blue, and then underneath the dura mater I've done a green arachnoid layer with fibers hanging down. That's why they call it the arachnoid layer, because this looks like a cobweb. And then they're attached, these fibers from the arachnoid, to the pia mater, which hugs the brain tightly. I illustrated this in yellow so you can kind of see it. Hopefully you can see it better at home when you're watching this. And it's beautifully illustrated in your textbook as well. Now, from the outside going in, as you know, the outside of the brain illustrates the skin, and hopefully we have plenty of hair on top of that skin. The next layer right here, this next layer is the skull itself. And you can see the compact bone on the outside layer of the skull and the inside layer of the skull. And then in the middle is the spongy bone, which carries on, if you remember correctly, the hematopoietic process, that process in making all those wonderful red blood cells and white blood cells and platelets right there in the spongy bone or the red bone or the myeloid. Now that's the cranium, thick cranial vault to protect the brain, as well as the vertebra which surround the descending spinal cord. So it protects it. And we also know that Mother Nature's given us that cerebral spinal fluid, which is coming from the choroid plexus inside the ventricles. And that choroid plexus, which is a ruffling of the blood vessels, which extracts from the blood the contents of the cerebral spinal fluid. And that fluid cushions the brain. If we jump up and down, the brain is going to move a little bit, but it's not going to smash against the cranium with a great deal of force, because it has that cushion, that cerebral spinal fluid that cushions it. If it wasn't for the fluid and the meninges, the brain would slam against the inside of the cranium and possibly break tiny capillaries. If the capillaries break and actually run amok amongst the brain tissue, they can do lots of damage. Direct blood in the soaking up the outside of the brain cells, that direct blood contains hormones and some amino acids and other toxins that can cause seizures. So we have to protect the brain from the blood inside the capillaries, even though that blood feeds them through the capillary wall. And that is done because of the meninges and this wonderful cerebral spinal fluid that bathes the brain and the spinal cord mater. It's very, very durable. It's very white in color, and it has the consistency of a white balloon. You know, when the air is out of the balloon, that white balloon has a lot of tenacity, a lot of strength. That's somewhat the way that dura mater is. It's difficult to puncture it. It's extremely protective. Now, one thing I want to mention about the dura mater is that it is one membrane, but has two layers. There's a layer that clings to the inside of the skull. They call that the periosteal dura mater. And there's a membrane that clings to the arachnoid layer, which is underneath, known as the meningeal layer. Now, these two layers of the dura mater, even though it's one membrane, two layers, they're important because in the brain, as you can see right here, the layer splits in some areas. And where they split, there is a villi, which we had referred to earlier as the arachnoid villi. And these arachnoid villi occur in the space between the split dura mater, the periosteal dura mater, and the meningeal dura mater. And where these villi punch out or move out from the arachnoid layer, what they do is they absorb in this sinus consistently, continuously absorb cerebral spinal fluid. So that is where the fluid is taken back, the cerebral spinal fluid, back into the bloodstream, where in the ventricles, the choroid plexus, which was the ruffling out a concentration of capillaries. Those concentrations of capillaries in that choroid plexus within the ventricles, that area is where the cerebral spinal fluid is made and then sent into the ventricles. Remembering that the cerebral spinal fluid made by those, which extract from the plasma of the blood and make the cerebral spinal fluid to secrete it into the ventricles so that they can go from the first and second lateral ventricles to the third to the fourth, around the entire brain with that cerebral spinal fluid and around the spinal cord, containing the ependymal cells, sending in to the ventricles the cerebral spinal fluid, and the arachnoid villus in these sinuses, in between the split dura mater. They reabsorb the old cerebral spinal fluid back into the bloodstream. So you're constantly making a fresh supply of cerebral spinal fluid, and you're constantly reabsorbing it in the tree-like area of the arachnoid villi. There are little capillaries that absorb the cerebral spinal fluid. But that is, and here the artist has illustrated it in light blue, is the arachnoid layer. Now, they call it the arachnoid layer because this meninges hugs the innermost dura mater, and hanging from the arachnoid mater or the arachnoid layer are these tiny cob-like fibers. These fibers hang down and attach to the pia mater, which is the innermost layer that actually hugs the gyri and sulci. Now, the importance of this arachnoid layer and these little fibers that are hanging down from that arachnoid layer, the importance of these fibers is that there is a space between the arachnoid and the pia mater, and this space is where cerebral spinal fluid flows. It flows amongst all of these fibers, and that's where the cushioning process occurs. That watery cushion of the cerebral spinal fluid in between the arachnoid layer and the pia mater in what we call the arachnoid space. And I illustrated it here. If I can turn it sideways. There. Here is the dura mater. The green is the arachnoid layer, and these fibers hanging down attach to the pia mater, and the cerebral spinal fluid flows amongst the subarachnoid space. Now they call it subarachnoid, sub meaning below the arachnoid layer. That's what that means. Now, these fibers, and you can see them beautifully illustrated here, and they are also illustrated in your textbook on page 390, and you can see those fibers hanging down from the arachnoid layer and directly attaching to the pia mater of the brain. It also shows you in this diagram the arachnoid villi in the sinuses where the dura mater is split. Okay. Now, the third layer is the pia mater. And in this illustration, the artist has shown a little white line that is hugging the gyri and dipping right down into the sulci, up to the gyri and sulci. When you look at a fresh brain that has been taken out of a body and you remove the cranium and the dura mater and you expose the fresh brain looks very, very slick and shiny and wet. The wetness comes from the fact that the outside of the brain is soaked in that cerebral spinal fluid. That's the wetness. And the shininess comes from that pia mater. Pia mater in Latin means soft mother because it hugs the soft gyri and sulci the convolutions of your brain. Inside this pia mater are thousands of tiny capillaries and blood vessels that actually feed the brain its nutrients. The brain receives its nutrients not only from the capillaries in the pia mater, but also the nutrition-rich cerebral spinal fluid that bathes your brain. Now, these capillaries in the pia mater are protected by what we will call later the, let me think, I just had a brain, I won't say it. The capillaries are protected by the blood-brain barrier, but the capillaries also use that same mechanism of feeding tissue, just as we explained earlier in the semester when we talked about a large artery breaking down to a smaller arteriole and then to the capillary. And at the capillary level, all the tissues of your body are fed. Do you remember our discussion of the arterial end of the capillary? And at the arterial end of the capillary, the hydrostatic pressure that builds up from the narrowing of the lumen, from the artery to the arteriole to the capillary, as it narrows the lumen, hydrostatic. So when it gets to the arterial end of the capillary, everything that is small enough to leave the capillary wall, some galactose and fatty acids and minerals and vitamins and minerals and hormones and enzymes, all of that leaves the arterial end of the capillary, flooding the tissues and feeding the tissues. Then on the venual end of the capillary, when the blood pressure has slowed down on the venual end, then reabsorption of materials re-enter the venual end of the capillary, such as nitrogenous wastes that is a byproduct of the metabolism of your body cells, urea, uric acid, creatinine, ammonia, excess salts, and enzymes and hormones not utilized. It'll come back in the venual end of the capillary. That happens with hydrostatic pressure in the capillary. But the capillaries that are in the pia mater are unique. They behave a little differently than the capillaries in the rest of your body. The capillaries that are in the pia mater, these capillaries have membranes. The capillary is bordered by a membrane. And remember, the capillary is small enough for one red blood cell, one white blood cell, and platelets to flow through the capillary in line. And as they do, the pressure, the blood pressure forces substances out. Well, in the brain, the pores in the capillaries are not as open and as numerous as the pores in the capillaries of the rest of your body. The capillaries in the brain, inside that pia mater, they have much tighter junctions. This happens starts to happen by the end of the first year of life. But they have much, much tighter junctions. And these junctions don't permit everything to go through the capillary wall that's small enough to go through that wall, as does happen. Some of those constituents, if they got out through the capillary wall of the capillaries in the pia mater and came in direct contact with nervous tissue, that cerebral cortex, which is millions and millions of cell bodies and dendrites, some of the hormones, some of the amino acids, and some of the enzymes, if they got out and came into direct contact with the brain, then they would be toxic, could cause brain damage, and sometimes even seizures. So the junctions in the wall of the capillaries of the pia mater are much tighter than the junctions in the wall of the capillaries of the rest of the body. Now, that's part of what we call the blood-brain barrier. Now, another part of the blood-brain barrier has to do with neuroglia, special neuroglia. Now remember, we talked about two neuroglia in the peripheral nervous system, Schwann cells that are wrapped around the axons of motor neurons in the PNS. And of course, there's also some satellite cells, which are neuroglia and the satellite cells and the Schwann cells were neuroglia in the PNS. Then we mentioned that there are 4 neuroglia in the central nervous system. The central nervous system have 4. And we mentioned the oligodendrocytes. That was one I talked about. Now, this, I think I've shown you, it's one of my favorite transparencies. And yes, it is on page 369 of your textbook. I love this illustration because it beautifully illustrates all 4 of these neuroglia in the brain. And of course, these neuroglia are in the brain, they're in the spinal cord too, and they're highly protective of the nervous tissue in the brain and the cord. We talked about the oligodendrocytes, and here, if you remember correctly, here is an illustration of the oligodendrocyte in the brain. Here is the nucleus and the cell body. If you can see that, it's in yellow. And the arms of the cell body, they reach the axon of this motor neuron, wrap around it and protect it. This arm wraps around another axon and protects it. This oligodendrocyte wraps around the lower portion of the axon, and this one, the lower portion. So the oligodendrocytes wrap around, they have extensions from their cell bodies that actually wrap around the axons in the cerebral cortex of your brain, protecting it. Now, we also mentioned microglial cells here. These are wandering cells around the brain that are kind of what we call the sanitation. Any enemies that get in the brain, protozoans or bacteria, they move around through the brain cells, just like white blood cells can move around through your tissues and phagocytize the enemy. Those are microglial cells. Here's one here and one here. Now, we were talking about the blood-brain barrier. So here is the neuroglia I want to mention that is the most important neuroglia in the blood-brain barrier. Here is your capillary. The artist has shown a little bit of the capillary wall here. And you see extensions from this astrocyte. Cyte, C-Y-T-E, means cell. Astro, it looks kind of like a star. So that's why they call it an astrocyte. Looks like a star. So the extensions from the cell body of the astrocyte wraps around the cell body that wraps around the axon itself. These extensions from the astrocytes wrap around and protect the capillaries. So the astrocytes, along with the tight junctions in the capillary membrane, blood-brain barrier, the BBB, and it begins to form in a baby around the end of the first year of life. The danger up until that point, and that is 12 months plus the gestational period, 9 months, the danger is in the developing embryo and fetus and the growth of the baby to the first year of life. It does not have this blood-brain barrier. It is in the process of developing it, but it doesn't have it. That's why we have laws about the consumption of alcohol and the taking of drugs during pregnancy, because alcohol, which is extremely toxic to the baby's brain, and the drugs, they can actually go through the capillary, damage. Okay. And so that's the reason, even when you pick up a bottle of wine or hard liquor on the label, it has the Surgeon General's warning, because they're warning you that any consumption by a mother during those gestational stages can actually do permanent damage to her fetus. And so we have to be extremely careful. And that's the same thing with any kind of prescription drug. If a woman's pregnant, she's always encouraged to read all the labels carefully on any prescription drug she may have so that she doesn't take in a drug into her body that can go directly into the brain of the fetus and cause damage. So that's the reason for those laws. And the laws are so strict that we have actually taken women who were pregnant that were alcoholics and continued to drink during that pregnancy. We have taken them into custody, and I don't want to say put them in jail, but they put them into protective custody during those gestational months so that she doesn't do damage. The baby is born. If the damage is done by the mother imbibing in all this alcohol, then that baby is born with fetal alcohol syndrome. And there are certain characteristics that are very obvious in a newborn and a young baby that definitely illustrates the characteristics of fetal alcohol, FAC, fetal alcohol syndrome. And you can probably Google that and look at some YouTube videos of the appearance of babies that have had mothers that took in alcohol during the pregnancy or illicit drugs. And we're talking about any kind of illicit drug, that cocaine, good grief, I can't even think of other ones, but heroin, all of these illicit drugs that damage the baby's brain. And that is the blood-brain barrier. Okay. Now, what can I ask you? If I call you on the phone and ask you to give me some answers, I can say, give me from the outside going in. That means starting from the skull, going to the actual cerebral cortex, the meninges of the brain. And of course, you could say the dura mater, the arachnoid layer, and the pia mater. I can ask you, what is the function of the pia mater? You could say the function of the pia mater is to clean capillaries that actually feed that cerebral cortex its nutrients. And these capillaries of the pia mater have tight junctions that won't permit certain substances to leave the capillary and come into direct contact with these vital neurons of the cerebral cortex. In addition, that blood-brain barrier would not only be the tight junctions of the capillaries, the pia mater, but also the astrocytes that protect the capillary wall. What would be is the middle layer. It's just underneath the dura mater. And this arachnoid layer has fibers that come down from that layer underneath the dura mater, and these fibers connect with the pia mater. The space that these fibers exist in is known as the subarachnoid space, through which the cerebral spinal fluid flows. And that means the inside of the cranium. That part of the dura mater is known as the peristeel dura mater. And there are two layers in that one membrane known as the dura mater, the peristeel layer, and then the meningeal layer. And I did mention too that in the brain, now this doesn't occur in the spinal cord, even though the dura mater, the arachnoid, and the pia mater are there. You don't have these arachnoid spaces or this split right here. Now this is a sinus that's different from the subarachnoid space. This is a split in the dura mater membrane, and it contains the fluid, cerebral spinal fluid, old cerebral spinal fluid, and these little villi coming from the arachnoid layer make villi, they call them arachnoid villi, or little pedunculated polyps that are filled with blood vessels, and they reabsorb the old cerebral spinal fluid. And so the dura mater helps to reabsorb this cerebral spinal fluid by accommodating the arachnoid villi. And it is also a very, very heavy, heavy, heavy layer, dura mater, that protects the brain from from puncture. Now, just as I mentioned the arachnoid villi in the sinuses of the split dura mater, I want to mention to you another structure I did talk about. We discussed the, use my pen, we discussed the choroid plexus, which is, again, is part of the pia mater. And so the pia mater, with this choroid plexus and these ependymal cells in that choroid plexus, they're the tissues that extract from the blood the contents of the cerebral spinal fluid, sending it into the ventricles. So lining these ventricles is the pia mater, and this ruffling, that red tissue, which is just a concentration of blood vessels, is the choroid plexus. Thus, the choroid plexus of the pia mater, with the help of the ependymal cells, makes the cerebral spinal fluid. And the arachnoid villi, and the artist has shown you the arachnoid villi here on the outside of the brain, the arachnoid villi, they reabsorb back into the bloodstream the old cerebral spinal fluid. The fresh is coming from the choroid plexus of the pia mater, and the old cerebral spinal fluid is being reabsorbed by the arachnoid villi of the arachnoid layer. Now, one thing I want to mention to you that's kind of interesting, our terminologies come to us from the Greeks and the Romans. They did it all. And the word arachnoid comes from the word arachne. And arachne comes from the Greek and Roman mythologies. They had gods and goddesses up on Mount Olympus who did all these wonderful things that were so interesting. But one of the goddesses, one of the minor goddesses, was Arachne, Arachne. And Arachne boasted constantly about her abilities to weave and embroider beautiful material, and that she boasted that she, of all the goddesses, could weave the most beautiful materials on Mount Olympus. Well, a goddess above Arachne was Athena. And Athena warned Arachne that it was unbecoming to boast about her weaving skills, and told her that she would suffer dire consequences if she continued to boast about her weaving skills. Arachne ignored this, continued to boast about her, and Arachne splintered into billions and billions of spiders that would climb around on the terrestrial form of the planet, and would weave cobwebs for eternity. So the presence of the cobweb and Arachne being condemned to the earth is where the Romans and Greeks got the word arachnoid layer, because of these fibers that hang down from the arachnoid membrane just underneath. Blood-brain barrier, I expect you to know that there's a structure and a function to it. The structure is the tight junctions of the walls of the membranes of the capillary, and the work of the astrocytes, which are one of the four that wrap around the capillaries and protect the capillaries from releasing any toxins into the brain that could destroy nerve tissue or neural tissue. Now, keep in mind too that the capillaries are releasing nutrients, the sugars and the vitamins and the minerals that are needed, but the tight junctions and the saves us, and that is the blood-brain barrier. Okay. While I mentioned that, I'm going to reiterate that is the astrocyte. I mentioned the microglia that wander throughout the nervous tissue and actually phagocyte that keep it from getting infected. The oligodendrocytes protect the axons. Don't forget that. These oligodendrocytes protect the axons. They do not protect the capillaries. The capillaries are protected by the astrocytes. And these stay in place where the microglia phagocytize the enemy moving throughout. Now, the fourth neuroglia is the ependymal cells, ependymal. And the ependymal cells are in the choroid plexus of the pia mater, and the artist has illustrated them right here. They extract within that choroid plexus the cerebral spinal fluid from the plasma and secrete it into the cavity of the ventricles, first, second, third, and fourth, and even down through the central canal. Now, these ependymal cells not only extract that cerebral spinal fluid from the plasma, they also have cilia that constantly move the cerebral spinal fluid from the first and second down to the third ventricle through that interventricular foramen, and from the third ventricle to the fourth by the cerebral aqueduct, then down around the brain, down around the spinal cord, and through the central canal of the spinal cord. Okay, that finishes lecture. You look at your study guide after lecture 16 in Neuro. We are You Need to Know, and 1 through 15 and various others from 15 on down that I have mentioned, you need to know. If I've mentioned those study guide questions, you need to know them and be prepared now to answer any questions if I call you and we can talk. I want you to know this material so that you can do well on the finals. That is it for today. I'm going to call some of you tonight, and hopefully I'll be able to get in touch with all of you at least